the topic that I like the most, telomeres. So I'll start with a short introduction of what telomeres are. So one of the fascinating facts about DNA is that on both strands, ends of it, one strand is longer than the other. This is fascinating just because we humans like symmetry, but there is something beautiful about these differences in length. So you can see that the ends of the chromosomes are composed of repeats of the form TTA, GGG, and they can span from 5 to 15 kilobases in length. And this part, which is longer, is called the G overhang, just because it contains a lot of G's here. And the presence of this G overhang allows DNA to form an interesting structure in the end, this loop-like structure. And the need for forming this loop-like structure is to protect the chromosome ends from being recognized as double-strand breaks, because otherwise some machinery in the cell would think that this is a break, and it would try to fuse two different chromosomes together, leading to chromosomal instability. However, the presence of this overhang comes to a cost to the cell, because every time the cell tries to divide and the DNA replicates, the replicative machinery cuts this piece to create this G overhang, and due to this cut, the telomeres will become shorter and shorter with each cell division. Usually when the telomeres get critically short, the cell has a mechanism to sense this, and usually they just stop dividing or they undergo programmed cell death. However, some of the cells will continue division. Probably these are the cells that contain some mutations in checkpoint proteins which allow them to bypass this telomeric crisis and continue the division, and they will lose the telomeric caps overall. Again, some of the cells will develop a mechanism whereby they either recover the cap of the telomere or do not allow it to be lost. And this will allow the cells to divide indefinitely, thus leading to tumor growth. This is one scenario by which cancer can start. Another scenario could be that some of the cells from the very beginning have activated this mechanism of telomere maintenance. So here, this abbreviation will be used, telomere maintenance mechanism. And if the cell activates this mechanism long before, then it will even not face this telomeric crisis, and this particular subset of cells will continue dividing, leading to tumor growth. These are very simplified and probably wrong models of how telomere regulation leads to cancer development. And in colon cancer, there are studies that have both implicated short and long telomeres in the risk of progression of the disease. So there is a variety of ways how telomeres can lead to cancer progression. Moreover, the telomere maintenance mechanisms themselves are heterogeneous. So there can be two pathways which can lead to this process. One of the pathways is utilized by expression of an enzyme called telomerase. So here you can see this is the enzyme complex, and the red one is a template, which is RNA. So RNA template contains a sequence complementary to the telomeric sequence, and the enzyme uses this template to elongate the short telomeres. An alternative pathway is utilized whenever this enzyme is not expressed. In this case, you again need a template to elongate the short telomeres, but you take the template from another chromosome's telomere. So homologous recombination events happen here, leading to telomere elongation. Currently, the factors which contribute to the decision of the cell to activate one pathway or another are not really known and not really well described. So we spent quite a lot of time on reading quite a bunch of papers to gather together and describe all the molecular events that are involved in both pathways. In an extended version, these pathways look like this, and they contain detailed information about protein-protein interactions that in a cascade of events lead to realization to telomere maintenance mechanisms. So the availability of these pathways allows us to implement a pathway signal flow algorithm, which basically takes gene expression values, maps them on to the nodes of this pathway, and here you can see red and blue nodes. So red means that this node is expressed in high amounts, blue means in low amounts, and using the information about protein-protein interactions, we can trace the flow of the signal from pathway inputs to their final outputs, and finally we can see which pathway is more active. So currently in this picture the alternative pathway is activated. Now why is this interesting? Why do we care about which pathway is activated in the cells? Mainly because we want to fight cancer. And cancer is fought currently with therapies, and there are multiple drugs currently on the market which try to inhibit the function of telomerase, the enzyme that elongates telomeres. Of course, because if these drugs work, then the cancer cells will not be able to divide indefinitely. Now, a lot of these drugs which affect telomerase, they fail during preclinical and clinical trials, and mostly because if you treat a tumor with a telomerase inhibitor, then it switches from telomerase pathway to the alternative elongation pathway. Or there can be cases where you actually treat a tumor which from the very beginning was utilizing this alternative pathway, and thus these telomerase inhibitors are not really working. There are also drugs which could affect the alternative elongation mechanism. However, their effect is currently not well described. Another interesting issue is to understand whether we have this simplistic binary classification of tumors which are either ALT or TEL. And there have been a couple of reports which say that coexistence of both pathways is possible in a single tumor. And it's a question of investigations to understand whether this is just a heterogeneity of cells within a tumor that utilize different pathways, or these pathways can coexist in the same cell at different levels. So with all these questions in mind, we proceeded to analysis of telomere regulation in colon cancer. Colon cancer is quite a heterogeneous disease. The University of Leipzig had data on a very small subset of colon cancer, which is its hereditary form, Lynch syndrome. And this colon cancer is dependent on mutations in certain genes, which are involved in a process called DNA mismatch repair. This is a process whereby DNA mutations in DNA are detected and repaired. And deficiency in this process can lead to appearance of increase in numbers of certain repetitive regions in the DNA, which we call microsatellites. So this cancer is described by a phenotype called microsatellite instability, and I will talk about mismatch repair in the next slide in more detail. So this is a hereditary form. However, there are other forms of colon cancer that are not familiar and are arising during the lifetime with acquired molecular changes, and this is why they are called sporadic. So one subtype of sporadic colorectal cancer also has this deficiency in DNA mismatch repair, and we downloaded data from TCGA public repository to also compare our analysis with this subtype. And as a control, we also took a subset of sporadic cancer, which is not microsatellite unstable, which does not have this deficiency in mismatch repair. So here you can shortly see the mismatch repair process. The DNA polymerase tries to replicate the DNA, it creates an insertion, and then the mismatch repair complex comes to rescue. Well, it appears that a number of proteins which are members of this mismatch repair pathway are also members of the alternative lengthening pathway of telomeres. And this is why we thought that mismatch repair would be something that would make telomere maintenance mechanism different in different types of cancer. And there are also different hints from different studies performed on a variety of cancers that tell that mismatch repair might affect telomere length regulation. So all in all, we had colon cancer described with mismatch repair deficiency and microsatellite instability, and we tried to understand whether that could affect telomere length regulation. That was the goal of this study. First of all, we downloaded whole genome sequencing data, DNA sequencing data, and used our software computer to measure the length of telomeres. And these are the first results that we got. So on these figures, you see Lynch syndrome and sporadic colorectal cancer. The green ones here are the healthy tissues taken from the same patient. And the blue ones are cancer, and the pink ones are adenoma. Here you see the same picture, but dark means cancer and light means healthy tissue. So you can see that the telomere length, this is MTL stands for mean telomere length, in this picture gets shorter in cancers systematically in all the data cycles that we studied. And if we look at the cell cycle regulation, we see that the more cells divide, the shorter the telomere. However, while low cell cycle values or low rates of cell division means that you can have diversity in the telomere length, once the cells start dividing intensively, the telomere length shortens, and then it stays at a certain threshold level. So this basically means that telomere shortening is an early event in carcinogenesis of colon cancer, and it already appears at the level of adenomas. The difference between cancer and reference was a bit higher in Lynch syndrome and in sporadic colorectal cancer, which was described by microsatellite instability. However, this difference, and actually there were studies that were reporting that mismatch repair could lead to more rapid attrition of telomeres previously. There is another fact that could lead to such difference in attrition rates. It's the patient's age. So actually, colon cancer, which is described with microsatellite instability, is diagnosed earlier than the microsatellite stable one, which means that the telomeres in the patients who are younger in the healthy tissues are longer. Thus, you have a more rapid attrition rate, and whether this is connected to mismatch repair or not is a matter of further investigations. And telomeres are actually not purely consisting of TTH-GG repeats, the canonical repeats. You can see repeats which have different varieties. So these are repeat variants which can be insertion, deletion, or substitution variants. And different reports tell that alternative pathway of telomere maintenance could be linked to generation of additional repeat variants. Our software also allowed us to measure these repeat variants, and here you can see a cartoon of a chromosome. These are the reference tissues on the top row, and this is cancer on the second row. So in green one are the canonical repeats, and all the varieties of variants are depicted in different colors. So you can see that we have shortening of both canonical stretches of telomeres as well as the repeat variants. And shortening in the repeat variant region was slightly lower than in the canonical regions. So this could mean two things. Either you have generation of new repeat variants in the cancer, or the canonical repeats get shorter much faster than the telomeric repeat variants. So why this can be the case? If you think about chromosomes, the telomeres do not start abruptly. They start gradually. So gradually you have certain telomeric repeats in different parts of chromosomes distributed differently, and then this part becomes more and more consisting only of pure canonical telomeric repeats. So this is a proximity effect, meaning that telomeric repeat variants are accumulated closer to the centromeres. So if you start shortening your telomeres, you will more affect the canonical parts than the repeat variants. So this led us to conclude that we do not have evidence to tell that there is a generation of new telomeric repeat variants in our datasets. So generally, to conclude the telomere dynamics part of this study, we think that telomere shortening is an early event in colon cancer carcinogenesis. There is no strict difference between different subtypes of colon cancer in regard to telomere length dynamics, and this model of telomere regulation in cancers is more applicable to our case. So, telomere length dynamics is not different between subtypes, but probably the mechanisms or the processes regulating maintenance can differ. So, we proceeded to RNA sequencing data analysis. You have seen these pathways again. You don't need to go into details here. But we have taken gene expression data from different samples, mapped them on top of these pathways, and performed the pathway signal flow analysis that you have seen already. And these are generally the results. On these plots, on the y-axis, you see the activity of the alternative pathway. On the x-axis is the activity of the telomerase pathway. Green ones are healthy tissues, and the rest of the colors are cancer or adenoma samples taken from the same patient. So you can really see that the activity of both pathways increases in all cancer samples that we have analyzed. And one interesting factor here is that it increases in both directions. So there is no clear-cut separation of samples which are either utilizing the telomerase pathway or those utilizing the alternative pathway. The box plot depicts the same picture. So this is the activity of the telomerase pathway, and this is the activity of the alternative pathway. And we see that this difference between cancer and reference tissue is statistically significant and a little bit more pronounced for the telomerase pathway here. Now, if we compare to sporadic cancers, here the green one again is the healthy tissue, and the red is the sporadic colorectal cancer, which is not described by microsatellite instability, so it's stable. And this one is described by microsatellite instability. So what we are seeing here is that there is an increase of telomerase maintenance pathway in the microsatellite stable cases. In microsatellite unstable cases, we again see an increase of the telomere maintenance pathway. However, there is a difference here. The variance of the telomerase pathway activity is much lower in these samples. If we look at the box plots, we see that the telomerase pathway activity goes very high in the microsatellite stable cases. The alternative pathway, on the other hand, is not activated in the microsatellite stable cases. You can see here that we observe activation of the alternative pathway only in a subset of microsatellite unstable cases. So generally, this allows us to conclude that we see some traces of alternative pathway activation in cancers which are described by microsatellite instability or deficiencies in the mismatch repair pathway. So next, we wanted to understand whether there are any genes which we can identify which are guilty for this difference. So for this, for example, if we wanted to look if gene number 3 in this pathway is responsible for these differences, we just neutralize its expression value. After neutralization, we compute the activity of this pathway, and we compare it to the original activity when the gene was left untouched. And this comparison can allow us to tell the effect of each gene on the overall pathway activity. So we call this the partial influence of each gene. Now I know that these details are not visible from far, but it's not really important. So here you can see on the y-axis the influence of each gene in Lynch syndrome and sporadic colorectal cancer for the telomerase pathway and for the alternative lengthening pathway. What we saw from this analysis is that in case of telomerase pathway, it was easy to identify 3 genes which have very high importance compared to the rest of the genes, and as a confirmation of the validity of this approach, these 3 genes were parts of the telomerase complex. An interesting thing is that TERT, which is the catalytic subunit of telomerase and is usually the target of telomerase inhibitors, the expression of TERT itself was not different, was different from cancer in cancer compared to the reference. However, it was not the most differentially expressed gene. So if we only looked at the TERT, which is usually looked at, we couldn't find it as guilty for the differences observed. However, if we look at the effect of TERT in the telomerase pathway, we see that it has a really big influence, thus highlighting the importance of considering the pathway topology, not only the expression of a single gene. And in case of the alternative pathway, you should also know that the scales are here different. So the influence of the gene is much lower than what you see here. So in case of alternative pathway, the genes had almost homogeneous effect values, which means that we couldn't identify a single or a couple of genes which were responsible for differential activation of the pathway. Okay, so next, to understand whether this pathway signal flow approach is valid or not, we needed an orthogonal method. And unfortunately, we didn't have an opportunity to do experimental analysis to find molecular hallmarks of pathway activation. So another thing that we undertook is to take this concept of guilt by association. So what guilt by association means is that if you have a certain set of genes which are correlated in their expression, it means that they are regulated in the same manner. And if a set of genes is regulated in the same manner, it can mean that they are together participating in a certain process. What you see here are each node is a gene or a process in our TMM pathway. And links are how much the expression of these nodes is correlated between each other. One thing, and the blue nodes highlight the telomerase pathway, while the red nodes highlight the alternative lengthening mechanism. And of course, one thing that you can see here, even though this looks like a radiculogram, is that all the blue and red nodes are highly interconnected. So what this can mean that there is co-regulation of the 2 pathways, and you can also see anti-correlation edges here. So there is one thing that we can conclude from this picture, and another thing is you can see here that telomerase pathway activity node is here, and the alternative pathway activity node is here. So it is quite far away and not connected. The alternative pathway node is not connected. And this is a case for microsatellite stable sporadic colorectal cancer. If we look at the centrality values of this node, the centrality value means how much each node is connected to the rest of the nodes in the pathway. We can see that the blue nodes have much higher centrality, are connected more than the red nodes. This basically allows us to conclude that the genes or the members of the telomerase pathway are more co-regulated in this particular case, which could tell us that there is probably more regulation of the telomerase pathway. And the reverse picture is seen for microsatellite unstable colorectal cancer, where if you look here, most of the nodes which have high centrality belong to the alternative pathway. So this is orthogonal explanation or hint that the alternative pathway is activated in the microsatellite unstable cases. So I have a couple of slides which explain why we undertook this complex analysis and not the traditional analysis, but I'm out of time, so I'll skip this part. So in summary, we analyzed different subsets of colorectal cancer, ones that are described with mismatch repair deficiency and ones that are not, and we find that there is a slight activation of the alternative pathway in the microsatellite unstable cases. And in case of Lynch syndrome, we see that there is a continuum of telomerase and alternative pathway activation and no clear-cut separation between cases. However, the activation of microsatellite unstable and stable sporadic colorectal cancer has different shapes. So one of the limitations of this study is the small number of samples that we had in case of Lynch syndrome, which is understandable because Lynch syndrome constitutes only about 30% of colorectal cases worldwide, and the data sets are not really accessible. Another thing is the absence of experimental hallmarks for telomere maintenance. There is currently no gold standard method to experimentally validate which pathway activation you have. They have identified different molecular hallmarks, like presence of certain bodies which are called APB bodies, or certain extra chromosomal telomeric sequences, and they find a correlation between these and the microscopic picture of the pathway activation. But there is no gold standard, and these methods are highly resource consuming. So we think that using gene expression data would allow to analyze much more samples and probably in a more accurate way. So next, I want to describe, put this whole study into the collaboration network that we have here. So this study started with a pathway signal flow algorithm in our absence group, and then continued to construction of these telomere maintenance pathways using this pathway signal flow algorithm and manual annotation of literature. Then we spent quite a lot of time on writing a paper with Dr. Hans Binder and his group, and currently it's in the process of revisions. During the next 2 years, I will probably look into the crosstalk between telomere maintenance mechanisms and other processes happening in the cell. So if you know, if you heard Vincent's talk yesterday, cells respond to the stress by regulating RNA quantities and RNA composition. So this process is called RNA surveillance in response to stress. So it appears that a lot of members of the genes which are involved in RNA surveillance are also involved in telomere maintenance, and we would like to investigate whether there is a systematic crosstalk between these two processes. And finally, I'll talk about the work of the next speaker, Rand. So they are working on biological text mining to extract interactions about protein-protein information about protein-protein interactions from biological texts. So instead of spending 3 years manually reading literature and trying to construct these TMM pathways, probably in the future, I don't really know how far in the future, we will be able to do this thing automatically and obtain much more extended and much more accurate pathways. Thank you.